Hey there, Mr. Mechnick, doing a little podcast on enzymes tonight, or today, this morning, this afternoon, whatever time you're watching it, doesn't really matter. Any time is a good time to learn about enzymes. So you ask, what are they? Well, they play a significant role in all biological systems. They are biological catalysts. So for you chemistry people, a catalyst is a chemical that can typically speed up the rate of a chemical reaction. We can identify an enzyme in this diagram as the substance that acts upon a specific substrate. So the enzyme acts upon a specific substrate. So this substrate can fit into the enzyme in an area called the active site. So when these two chemicals join together, it will allow a certain chemical reaction to take place. So just to give you the definitions of a substrate and the active site, the substrate is the molecule or molecules upon which an enzyme acts upon. And the active site is the part of the enzyme that fits with the substrate. It's the actual site that fit these two together. This is very important because every specific enzyme has a specific substrate that it can act on. Here's an example.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
So as you just saw, when the enzyme and the substrate combine together to allow for the chemical reaction to take place, you have what's known as an enzyme-substrate complex. The interaction between this particular protein and the chemical substance that is being reacted. Some common characteristics that you guys need to understand about enzymes are that they are very, very specific. Think of it as a key in a lock model. A specific key can only act on a specific lock. An enzyme can only act upon a specific substrate. As you are aware, enzymes are proteins. A common characteristic that we would see when we are reading about enzymes, or if you come across a word that ends in ase, you should recognize it as an enzyme. Some that we've already mentioned in class would include amylase. This is an enzyme that breaks down the amylose sugar. Lactase is another specific enzyme that breaks down lactose sugar. Other enzymes that you will hear about this semester and second semester are things like helicase, DNA polymerase, alcohol dehydrogenase, RNase, lipase, proteases. These are all different types of enzymes that act on specific substrates. Another characteristic about enzymes that are very important is that during the reaction, these enzymes do not get used up. They are not part of the reaction itself. They make the reaction happen, they make it go smoothly, but they are not actually chemicals used up during the reaction. They are not considered reactants, and they are not products of the reaction. The other big characteristic of enzymes is that they really allow chemical reactions to happen without maximum amount of energy required. They can decrease the amount of energy necessary to start the chemical reaction. So for instance, if you have an enzyme that needs to work on a substrate and you did not have the enzyme present, it would take a lot of energy in order for that reaction to take place. As you can see on this top dark line. When an enzyme is present, it lowers the amount of activation energy, and the reaction can happen much more quickly and smoothly when you have less activation energy, when you have that enzyme present. So the big feature is an enzyme will lower the activation energy. So since enzymes play a big role in biological systems and regulating chemical reactions, I would like you to consider factors that can affect the chemical reaction. What are some things that you might consider that could play a big role in affecting the rate of the chemical reaction when an enzyme is present? So please either Google search or just brainstorm and think of some factors that can affect the rate of a chemical reaction with an enzyme. Bring those factors to class tomorrow, and that will help us figure out a lab that we're going to be working on where we look at different factors that can influence the rate of a chemical reaction.